There's a rock in the Australian outback that shouldn't exist. It's 3,465,000,000 years old, nearly as old as the planet itself, and embedded inside it, visible only under a microscope, are structures that look exactly like bacteria. Filaments, chains of cells, the oldest fossils on Earth. Except for decades, no one could agree if they were actually fossils, or just tricks of chemistry, minerals masquerading as life. The debate turned into a scientific war that lasted 30 years, until someone found a way to prove, definitively, that these microscopic threads were once alive. But here's what should terrify you. If life existed 3.5 billion years ago, when Earth was barely cooled from its formation, then life didn't slowly emerge over eons. It exploded, almost instantly. Which means one of two things. Either we're looking at this completely wrong, or life is far more inevitable, more unstoppable than anyone ever imagined. Before we dive in, please consider subscribing and liking this video. Your support truly helps us continue this work. Thank you. Tracking down the earliest fossils has taken nearly 2 centuries of paleontological discovery. Scientists have scoured the most remote and inhospitable places on the planet, developed entirely new analytical techniques, all in a singular quest to answer one of the most profound questions in science. When did life begin? And what evidence remains? What began with Mary Anning braving English storms to make a living from selling stones continues to this day. Except now, the hunt has taken us to the Australian outback, the frozen wastes of Greenland, the wilds of northern Canada, searching for fossils so old, so altered by time, that they're barely distinguishable from the rock that entombs them. This is the story of the oldest fossils on Earth, and the scientists who risked everything to find them. Mary Anning's job, difficult as it was, had certain advantages. The fossils she sought were large, well preserved, and relatively abundant in the limestone cliffs near her home. When she uncovered a fossil skull, like other references to colossal monsters occasionally pulled from the earth in antiquity, it was unmistakably a skull, albeit of some mysterious extinct creature. The bones looked like bones. The teeth looked like teeth. You could see what you were looking at. But the deeper in time paleontologists probe, the harder their task becomes exponentially. The further back in the fossil record we go, the more alien the creatures become. Past 500 million years ago, life becomes dramatically simpler. No bones, no teeth, no shells at first, just soft-bodied organisms that rarely fossilize. And before that, microscopic life, single cells, bacteria, organisms so simple they can be confused with mineral formations, with crystals, with specks of carbon that form through purely chemical processes. So if we're going to search for the oldest fossils on Earth, fossils from billions of years before Mary Anning's ichthyosaurs, we need to understand exactly how something living becomes stone, how an organism is preserved across geological time. Because the odds, it turns out, are spectacularly bad. When any living organism dies, biological and chemical agents move quickly to devour it. Fungi, bacteria, scavengers. They recycle the precious carbon and nutrients, breaking down soft tissues within days, sometimes hours, leaving nothing behind. For creatures with hard parts, bones, teeth, shells made from minerals like calcium carbonate or phosphate, decay is slower. But even these mineralized structures eventually dissolve, break down, return to the soil. The great cycle of life is ruthlessly efficient. Nothing is wasted. For an organism to be preserved as a fossil, it must be entombed, sealed away from scavengers and decay before they have a chance to act. And that means burial, fast burial. Organisms that die and fall into sediments accumulating at the bottom of oceans, lakes, rivers, swamps, these have the best chance. Surrounded by mud and sand, cut off from oxygen, isolated from most biological activity, the remains are protected. And then, slowly, as more sediment piles on top, as geological forces compress and transform loose sand and mud into solid rock, any organic structures within become templates, molds, spaces where minerals can precipitate, replacing the original material atom by atom, preserving the shape of the long-dead organism as rock within rock. Petrification, turning to stone. In this way, the remains of verdant forests millions of years old can be seen all over the earth. Petrified wood, tree trunks turned to solid silica or calcite, preserving every detail of the original cellular structure. It's beautiful, and it's rare. Because despite their apparent permanence, these rock repositories are not eternal. Over the vast stretches of geological time, tectonic forces shift entire landscapes. Continents collide, oceans open and close, rocks are folded, faulted, lifted into mountain ranges miles high, or plunged deep into the crust. The rocks, and any fossils within them, are crushed, heated, chemically altered, often beyond recognition. Sometimes they're destroyed completely, subducted at plate boundaries, melted back into magma, or shattered and eroded away when exposed at the surface. The older a rock is, the more of these tectonic convulsions it has witnessed over the eons, and the more likely it is to be altered or destroyed. In fact, rocks older than about 3 billion years are vanishingly rare on Earth. Most of the planet's ancient crust has been recycled, consumed, erased. So the odds of finding a fossil of the earliest life on Earth are almost incomprehensibly small. Think about it. That organism, whatever it was, had to avoid biological decay without any mineralized shell or hard parts. Its soft, organic matter had to be buried within hours. The right minerals had to be present in the sediment to preserve delicate microscopic structures. Then the rock had to form and survive billions of years of tectonic chaos without being heated, squeezed, or eroded beyond recognition. One organism in a trillion becomes a fossil, and of those fossils, only a handful survive to the present when the rocks are young. For rocks 3.5 billion years old, the odds become astronomical. It's like finding a specific grain of sand on a beach, except the beach is the size of Australia, and the sand has been through a furnace multiple times. And yet, scientists have found them. Against all probability, we have fossils from the earliest epochs of Earth's history. Fossils that document life nearly 3.5 billion years ago, when our planet was barely 1 billion years old. But finding them required going to one of the most remote and inhospitable places on the planet, the Australian outback, Western Australia, a place called North Pole, named by 20th century miners who once toiled here, a darkly ironic reflection of its stark remoteness. But in every other respect, this patch of the Australian desert could not be further removed from its Arctic namesake. This is one of the hottest, driest, most unforgiving landscapes on Earth. A desolate expanse of red dirt and stone, studded with rounded tufts of spinifex grass that appear innocuous but are laden with microscopic silica needles. Touch them, and they'll rip through your clothes and embed in your skin like fiberglass. Fieldwork here is genuinely dangerous. Geologists working at North Pole rarely wander far from their reinforced four-wheel drive vehicles. There are no roads out here, no cell service, no water. Temperatures regularly exceed 50 degrees Celsius in the shade, except there is no shade. The sun beats down mercilessly. This is rattlesnake and redback spider territory. It's easy to die out here if you're not careful. And yet, for paleontologists, this brutal landscape is a gift, because with so little water and vegetation, the rocks are completely exposed, naked, unweathered. You can see the geological structures clearly, trace individual rock layers for kilometers, sample exactly what you need without having to dig. The desert preserves what rainforests and grasslands hide. And among the rocks exposed here at North Pole are some that are extraordinarily special. They're called the Apex Chert. Dark-colored, glassy rocks made almost entirely of silica, with just a trace of carbon trapped inside. They formed from a kind of gel that accumulated on an ancient seafloor under conditions very different from today. The surrounding rocks help reconstruct the environment. Volcanic rocks, pillow basalts, formed when lava erupted underwater. Sedimentary layers that indicate shallow marine conditions. This was an ancient ocean, dotted with volcanic islands. The chert formed on the seafloor of this primordial sea in the presence of hot, mineral-rich hydrothermal fluids circulating upward from magma chambers deep in the crust. Like the black smokers we find on mid-ocean ridges today, except this was happening 3.5 billion years ago. We know the precise age because geologists can measure the isotopic ratios of elements in the volcanic rocks. Radiometric dating. The decay of radioactive uranium into lead, rubidium into strontium. These atomic clocks tell us exactly when the rocks crystallized from molten magma. And the answer for the Apex Chert and its surrounding volcanic layers is 3,465 million years old. Let that sink in. 3.465 billion years. The Earth itself is only 4.54 billion years old. These rocks formed when our planet was barely 1 billion years into its existence. When the moon was closer, the days were shorter, the sun was dimmer, the atmosphere had no oxygen. An alien world. The age and preservation of the Apex Chert are remarkable. But that's not what makes these rocks famous. Because embedded in the chert, visible only under powerful microscopes, are tiny structures, filaments, tubes, hollow spheres, made from specks of black carbon, each 1/100th of a millimeter across, sometimes arranged in chains. And they look, for all the world, like bacteria. Ancient fossilized bacteria. The oldest direct evidence of life on Earth. But here's where the story gets complicated. Because proving that these structures are actually fossils and not just unusual mineral formations turned into one of the most contentious scientific battles of the last 30 years. A battle that's still not entirely resolved. And it all started with one man working in a laboratory at UCLA in the early 1990s. His name was Bill Schopf, and he was about to make the discovery of a lifetime. William Schopf was already a respected paleontologist by the early 1980s. He'd spent years studying Precambrian rocks, the vast stretch of Earth's history before complex life appeared 540 million years ago. Precambrian fossils were notoriously difficult to find and interpret. Most scientists avoided the field entirely. Too hard, too controversial. But Schopf was drawn to it precisely because it was difficult. In 1982, he travelled to Western Australia specifically to collect samples from the Apex Chert. Other geologists had noted these ancient rocks, but no one had examined them closely for fossils. Schopf spent weeks in the field, enduring the punishing heat, carefully collecting samples from outcrops scattered across the North Pole region. He packed them carefully and shipped them back to his lab at UCLA. And then he waited. Because analyzing rocks this old requires patience, thin sections had to be prepared. Slices of rock, ground down until they were only 30 microns thick, thin enough to transmit light under a microscope. Each section took hours to prepare, and the Apex Chert was hard, silica, essentially glass. It destroyed saw blades and grinding wheels. But when Schopf finally placed the first thin section under his microscope and adjusted the focus, he saw something that made his heart race. Filaments, delicate thread-like structures, darker than the surrounding rock. Organic carbon. And they weren't random. They had form, structure. Some were segmented, divided into cells like modern filamentous bacteria. Others were smooth, unbranched. Still others formed chains, or clusters, or hollow spheres. Over the following months, Schopf catalogued 11 distinct morphological types, 11 different forms, each consistent within itself, each distinct from the others, and crucially, each resembling modern microorganisms. Cyanobacteria, the photosynthetic bacteria that produce oxygen today, have filamentous forms almost identical to what Schopf was seeing in 3.5 billion-year-old rock. In April, Schopf published his findings in the journal Science. The paper was titled Microfossils of the Early Archean Apex Chert, New Evidence of the Antiquity of Life. It was careful, rigorous, detailed, and it made an extraordinary claim. Life on Earth was at least 3.465 billion years old. Not just life, but diverse life, complex enough to have evolved into multiple distinct forms, sophisticated enough to potentially photosynthesize, producing oxygen. The scientific community was stunned. If Schopf was right, life hadn't just appeared early in Earth's history. It had exploded, diversified, thrived, within the first billion years of the planet's existence. The implications were staggering. It suggested that life wasn't a fluke, that given the right conditions, simple chemistry became biology quickly, inevitably perhaps. For nearly a decade, Schopf's discovery was accepted. The Apex microfossils became textbook examples, the oldest fossils on Earth, evidence that life arose fast and flourished early. Museums built exhibits around them. Other scientists cited them as proof that the early Earth was more hospitable than anyone had imagined. And then, in 2002, a soft-spoken British paleontologist named Martin Brasier stood up at a scientific conference and said, very politely, that he thought Bill Schopf was wrong. Martin Brasier was a professor at Oxford University. Like Schopf, he specialized in Precambrian paleontology, the study of early life. But Brasier was a skeptic by nature. He believed that extraordinary claims required extraordinary evidence. And when he examined Schopf's data, read the papers carefully, looked at the images, he started to see problems. The filaments in the Apex Chert, Brasier argued, weren't necessarily biological. They could be explained by entirely abiotic processes, non-living chemistry. He pointed out that carbon can form filament-like structures during certain types of mineral growth. When silica crystallizes from a gel, which is exactly how chert forms, It can push carbon impurities along the edges of growing crystals. Where multiple crystals meet, the carbon concentrates, forming lines, networks, structures that can look remarkably like fossil microbes. And the shapes themselves, Brasier continued, were suspiciously simple: tubes, spheres, chains. These aren't complex morphologies. They're some of the most basic geometric forms that can arise from physical processes. Bubbles form spheres. Cracks form lines. You don't need biology to make a tube. Furthermore, Brasier noted, the Apex Chert had been heated and deformed since it formed. Metamorphosed, geologists call it. Not severely, but enough to alter the original textures. Enough to stretch and distort any structures inside. Could Schopf's fossils just be stretched mineral artifacts? Distorted carbon particles that only looked like cells because we wanted them to look like cells. In 2002, Brasier published his critique in the journal Nature. The response was immediate and fierce. Schopf fired back, defending his interpretation, pointing to the consistency of the structures, their resemblance to modern organisms, their occurrence in the multiple samples from multiple locations. This wasn't just one odd structure. This was a whole population, a community. But Brasier wouldn't back down. And over the following years, the debate intensified. Papers were published, rebuttals written, conferences became battlegrounds. In 2011, the Geological organized a formal debate, Schopf versus Brasier, in front of an audience of their peers. It was unprecedented. Scientific controversies are usually resolved quietly through data and peer review. But this was too important, too fundamental. The oldest fossils on Earth were at stake. Both sides presented their evidence. Schopf showed his images, his measurements, his comparisons to modern bacteria. Brasier countered with chemical analyses, experiments showing how carbon could be redistributed during metamorphism. Alternative explanations for every feature Schopf claimed was biological. At the end, there was no clear winner. The audience was divided. Some were convinced by Schopf, others sided with Brasier. Many were simply unsure. The debate had reached a stalemate. And then, science did what science does best. It moved beyond personalities and looked for new evidence. Because there was something both Schopf and Brasier had overlooked, something that could potentially settle the argument. Not the shape of the structures, which could be debated endlessly, but their chemistry, specifically their carbon isotopes. Life, it turns out, is picky about its carbon. On Earth, carbon comes in two stable forms, two isotopes with different numbers of neutrons in their nuclei. Carbon-12 has 6 protons and 6 neutrons. Carbon-13 has 6 protons and 7 neutrons. That single extra neutron makes carbon-13 very slightly heavier than carbon-12. And living organisms, when they metabolize carbon, preferentially use the lighter isotope. Why? Because biochemical reactions are lazy. They take the path of least resistance. Enzymes that fix carbon from the atmosphere, like the ones used in photosynthesis, discriminate against the heavier carbon-13. It's fractionally harder to move, fractionally slower to react. So organisms preferentially incorporate carbon-12 into their cells. This means that organic matter produced by life has a distinctive isotopic signature. It's depleted in carbon-13 compared to inorganic carbon in the environment. Not by much, a few percent, but it's measurable, and it's a smoking gun. Because there are very few non-biological processes that fractionate carbon isotopes in the same way. If the structures in the Apex Chert were truly biological, their carbon should be isotopically light, depleted in carbon-13. If they were just mineral artifacts, their carbon should have the same isotopic composition as the surrounding rock. The problem was, measuring carbon isotopes in structures 1/100th of a millimeter across, embedded in rock 3.5 billion years old, is extraordinarily difficult. The technology didn't exist when Schopf and Brasier were arguing in 2002. But by 2010, it did. A technique called secondary ion mass spectrometry, SIMS, allowed scientists to measure isotopes in tiny spots just a few microns across. Essentially, you bombard the sample with a focused beam of ions, knock carbon atoms off the surface, and measure their mass with incredible precision. You can target individual microscopic structures, measure their isotopic composition, compare them to the surrounding rock. In 2018, John Valley, a geochemist at the University of Wisconsin, led a team that did exactly that. They took Schopf's original samples, prepared new thin sections, and analyzed the carbon isotopes in the filamentous structures using SIMS. The results were published in the Proceedings of the National Academy of Sciences. The filaments were isotopically light, depleted in carbon-13 by 37 parts per thousand compared to the surrounding rock. That's a huge fractionation, well within the range produced by biological carbon fixation. And crucially, the isotopic composition varied between different filament types, as if different organisms with different metabolisms were producing slightly different isotopic signatures. It was the smoking gun. The evidence that even Martin Brasier, who tragically died in a car accident in 2014, had said would be necessary to prove the structures were biological. The carbon isotopes strongly suggested that the Apex Chert filaments were not mineral artifacts. They were fossils. The preserved remains of microorganisms that lived 3,465,000,000 years ago. Today, the scientific consensus, though not unanimous, is that Bill Schopf was right. The Apex Chert contains the oldest fossils on Earth. But the story doesn't end there. Because even as the debate over the apex microfossils raged, other scientists were finding hints, tantalizing suggestions that life might be even older. Which raises the question, if we have fossils from 3.465 billion years ago, can we push deeper? Can we find evidence of life at 3.5 billion, 3.7, 4 billion years ago? The answer is complicated, because we're reaching the edge of what's possible, the edge of the knowable. Let me explain why. To find fossils older than the Apex Chert, we need rocks older than the Apex Chert, and those rocks are almost impossibly rare. Most of Earth's early crust has been destroyed, subducted, melted, recycled through plate tectonics over billions of years. The continents we have today are built on the wreckage of countless earlier continents that collided, merged, split apart, and sank back into the mantle. There are only a handful of places on Earth where rocks older than 3.5 billion years still exist, and even fewer where those rocks are in good enough condition to preserve fossils. Most have been metamorphosed, heated and squeezed until their original textures are obliterated, turned into gneiss, schist, other rocks so altered that any fossils they once contained are long gone. But there are a few exceptions, a few places where, by geological luck, ancient rocks have survived relatively intact, and paleontologists have been searching them for decades. One of those places is not far from North Pole in Western Australia, just a few dozen kilometers away, in fact, in a formation called the Dresser Formation. These rocks are slightly older than the Apex Chert, about 3.48 billion years old, only 20 or 30 million years older, which sounds like a lot, but in geological terms is nothing, a blink. The Dresser rocks are different from the Apex Chert. They're not made of silica. They're layered sedimentary rocks, sandstones and siltstones, the kind that form in shallow coastal environments, mudflats, tidal zones, and within these rocks, on the ancient surfaces that were once seafloor or tidal flats, there are textures, wrinkled patterns, tufted structures, bumps and ridges. And these textures look, for all the world, like microbial mats. The kind of mats formed by communities of bacteria living in modern estuaries and coastal zones. When bacteria grow on a sediment surface, they form sticky biofilms that trap sediment particles, creating layered structures with distinctive textures. And when those surfaces are buried and turned to rock, the textures are preserved. Fossilized, the structures in the Dresser Formation are compelling. Multiple research teams have studied them, and most agree they're probably biological in origin, probably formed by microbial mats. But there's a problem. While the textures are preserved, the organisms themselves are not. Unlike the Apex Chert, where silica gel entombed cells before they could decay, the Dresser rocks didn't provide that level of preservation. The sediments were deposited rapidly enough to make molds and casts of the mat structures, but conditions weren't right to preserve the microbes themselves. So we have evidence that life existed 3.48 billion years ago. Tentative evidence, suggestive evidence, but no actual cellular fossils, no direct proof. It's frustrating. We're so close, 20 million years older than Apex, but we can't quite cross the threshold into certainty. And then there's Greenland. In 2016, a team led by Alan Nutman from the University of Wollongong in Australia announced a discovery that stunned the paleontological community. They'd found stromatolites, those layered dome-shaped structures built by microbial mats, in rocks from Greenland. And not just any rocks, some of the oldest rocks on Earth. The Isotopes Greenstone Belt, 3.7 billion years old. The Isotopes rocks had been known for decades. They're famous among geologists because they're so ancient and, miraculously, they haven't been completely destroyed. But they're not in good shape. They've been metamorphosed, heated to over 400 degrees Celsius, folded and faulted. Most scientists assumed that any fossils they might have once contained were long gone, cooked out of existence. But Nutman's team found an outcrop, freshly exposed by melting snow, where the metamorphism wasn't as severe, where some of the original sedimentary structures had survived. And among them were cone-shaped layered structures up to 4 centimeters tall that looked remarkably like stromatolites. If true, this would push the fossil record back another 200 million years, to 3.7 billion years ago, just 800 million years after the Earth formed. When the planet was still being bombarded by asteroids, when the crust was barely solid. The implications were enormous. Life wasn't just early, it was immediate. As soon as the Earth cooled enough to have liquid water and a stable crust, life appeared, thrived, built structures large enough to see with the naked eye. But the announcement was met with immediate skepticism, because the structures, while suggestive, weren't definitive. Stromatolites can be mimicked by entirely non-biological processes. Sediments can be folded and deformed by currents, by tectonic forces, creating layered domes that look biological but aren't. And the ichthyosaur rocks, as ancient and altered as they are, have been through a lot. Could these structures be stromatolites? Maybe. But they could also be tectonic artifacts. In 2018, a rival team published a rebuttal in Nature, arguing that the ichthyosaur structures were not stromatolites, that they were just deformed sedimentary layers. The debate continues. Without better preservation, without additional evidence, we can't be sure. And that's the problem. As we push deeper into geological time, the evidence becomes more and more ambiguous. The rocks are more altered, the structures less clear. We're reaching the edge of what fossilization can preserve, and the edge of what we can confidently interpret. But there's one more piece of evidence, the oldest hint of life we have. And it doesn't come from a rock, it comes from a single crystal, a mineral grain. Smaller than a grain of sand. But it might contain the isotopic signature of life 4.1 billion years ago. It's called the Jack Hills Zircon, and it's the subject of the final part of our story. Zircon is a remarkable mineral. Chemically, it's zirconium silicate. But what makes it extraordinary isn't its chemistry, it's its durability. Zircon crystals are nearly indestructible. They resist heat, pressure, chemical weathering. While other minerals break down and dissolve, zircon persists. And because zircon incorporates uranium into its crystal structure when it forms, and uranium decays into lead at a known rate, each zircon crystal is a tiny atomic clock, telling us exactly when it crystallized. Most minerals don't survive the geological chaos that reshapes the Earth's crust over billions of years. They're melted, dissolved, crushed into powder. But zircon endures. And sometimes, when an ancient rock is finally destroyed, eroded away by wind and water, the zircon crystals it contained are released. They wash into rivers, settle into sediments, and get incorporated into new, younger rocks. Recycled, reborn, carrying their ancient age signature with them. This is what happened at a place called Jack Hills in Western Australia, about 800 kilometers south of North Pole. The rocks exposed at Jack Hills are metamorphosed sedimentary rocks, about 3.3 billion years old. Respectable age, but not unusual for this part of Australia. But when geologists examined these rocks closely in the 1980s and 90s, they found something extraordinary embedded within them. Zircon crystals, thousands of them. And when they measured the ages of these crystals using uranium-lead dating, they discovered that many of them were much older than the rocks containing them. Much older. Some were 3.7 billion years old, 3.9, 4 billion. And one crystal, a single grain analyzed by John Valley and his team at the University of Wisconsin in 2001, was 4.1 billion years old. 4.1 billion. That makes it the oldest piece of Earth ever found. Older than any rock, older than any mountain range. This tiny crystal formed when the Earth was only 450 million years old, less than 10% of its current age, when the moon was still glowing from the heat of the impact that created it. But here's what makes this crystal relevant to our story. Trapped inside it, incorporated into the crystal's structure as it grew 4 billion years ago, are tiny inclusions, specks of carbon, graphite, and that carbon has an isotopic signature. In 2015, Valley's team used advanced mass spectrometry to measure the carbon isotopes in the Jack Hills Zircon inclusions, and they found that the carbon was isotopically light, depleted in carbon-13 by about 24 parts per thousand. Not as much as the Apex Chert filaments, but still significant, still within the range that could indicate biological fractionation. The implication is staggering. If that carbon signature is biological, if it represents the metabolic activity of living organisms, then life existed on Earth 4.1 billion years ago, just 450 million years after the planet formed, when the surface was still being pummeled by asteroid impacts, when oceans were boiling away and reforming, when the crust was barely stable. But before we get carried away, we need to be honest about the limitations. This is one crystal, one tiny inclusion, one measurement. There are no cellular structures, no morphology to examine, no context, just an isotopic anomaly. And isotopic anomalies can be produced by non-biological processes, especially in rocks this old, this altered. Organic carbon from later contamination, perhaps, or unusual chemical conditions during metamorphism. Or carbon that was produced by abiotic processes that happen to fractionate isotopes in similar ways to biology. Hot water circulating through rocks can do strange things to carbon chemistry. So the Jack Hills Zircon is tantalizing, provocative, but not conclusive. It's a hint, a suggestion, a data point that makes us wonder. But it's not proof. Not the way the Apex Chert structures, with their morphology and consistent isotopic signatures and contextual environment, are proof. Still, the fact that we're even having this conversation is remarkable. We're talking about evidence for life 4 billion years ago, when the Earth was an infant. And that raises a profound question. How fast did life arise? Because here's what we know. The Earth formed 4.54 billion years ago. For the first few hundred million years, the surface was molten. Impacts were frequent and catastrophic. Any water that accumulated was vaporized repeatedly. This was the Hadean Eon, named after Hades, hell. No life could have survived on the surface during this time. But by about 4 billion years ago, maybe a bit earlier, the bombardment had diminished enough that liquid water could persist. Oceans formed, the crust stabilized, and within, at most, a few 100 million years, possibly much less, life appeared. If the Jack Hills carbon is biological, the window is even narrower. Life by 4.1 billion years ago means it arose almost as soon as the Earth was habitable, within the geological blink of an eye. And by 3.465 billion years ago, when the Apex Chert organisms were living, life wasn't just present. It was diverse, multiple distinct forms, complex enough to have evolved different metabolic strategies, possibly photosynthesizing, possibly producing oxygen, though the atmosphere wouldn't accumulate oxygen for another billion years. So what does this tell us? It tells us that life, at least on Earth, arises fast. Given liquid water, an energy source, and the right chemistry, simple molecules organize themselves into living systems almost immediately. Not over billions of years, over millions, possibly less. And that has profound implications for the question of life elsewhere. If life arose fast on Earth, it probably arose fast on Mars, which was Earth-like 4 billion years ago, with oceans and a thick atmosphere. It probably arose in the subsurface oceans of Europa and Enceladus, where liquid water and chemical energy exist today. It might arise on any of the billions of Earth-like planets we're discovering around other stars. Life, it seems, isn't a fluke. It's chemistry doing what chemistry does when conditions are right. And conditions are right more often than we used to think. But there's a dark side to this realization as well. Because if life arises easily, why haven't we found it elsewhere yet? Why is the universe so quiet? That's a question for another time. For now, we need to address one more controversial claim, the most contentious fossil discovery of the last decade, from the remote wilderness of northern Canada. In 2017, a team led by Dominic Papineau from University College London announced the discovery of what they claimed were the oldest fossils ever found. Not in Western Australia, not in Greenland, but in a place called the Nuvvuagittuq Greenstone Belt in northern Quebec. And the age they were claiming was almost unbelievable. Somewhere between 3.77 and 4.28 billion years old. The uncertainty in that age is itself a red flag. 500 million years of uncertainty is enormous. It reflects the fact that the Nuvvuagittuq rocks are extremely difficult to date. They've been so altered, so metamorphosed, that standard radiometric dating techniques give inconsistent results. Some methods suggest 3.77 billion years. Others suggest 4.28. And without a precise age, any fossils found within them exist in a kind of temporal limbo. But setting aside the age problem, what did Papineau's team actually find? They found structures, tiny tubular and filamentous forms made not of carbon but of iron minerals, hematite, magnetite, ranging from a few microns to 0.5 millimeter in length. And these structures were embedded in layers of jasper and chert that appeared to be deposits from ancient hydrothermal vents. Underwater volcanic systems, like the black smokers we find at mid-ocean ridges today. Modern hydrothermal vents support thriving ecosystems, bacteria and archaea that metabolize hydrogen, methane, sulfur compounds. And some of these microorganisms, particularly iron-oxidizing bacteria, produce filamentous structures made of iron minerals as a byproduct of their metabolism. They literally excrete tiny iron tubes as they grow. The Nuvvuagittuq structures looked remarkably similar to these modern iron-oxidizing bacteria. Same size, same morphology, same association with hydrothermal deposits. Papineau argued that they were fossils, the mineralized remains of ancient iron-metabolizing microbes that lived around vents on the early Earth. If true, and if the older age estimate is correct, these would be the oldest fossils on Earth by 800 million years. They would push the origin of life back to within 150 million years of Earth's formation. They would suggest that life arose not in shallow, sunlit seas, but in the pitch-black depths around volcanic vents, which would align with modern theories that place the origin of life in hydrothermal systems rather than surface ponds. But the reaction from the scientific community was swift and skeptical, because there are problems, big problems. First, the age. Without a precise date, we can't be sure these structures are older than the Apex Chert. They might be 3.77 billion years old, making them comparable to the Azure stromatolites, or they might be much younger. The uncertainty is too large to make definitive claims. Second, there's no organic carbon. Unlike the Apex Chert, where the fossils are made of carbon with biological isotopic signatures, The Nuvuagituk structures are made entirely of iron minerals. No carbon means no isotopic analysis, no chemical smoking gun. We can't test whether these structures were produced by biology or by abiotic chemistry. And third, iron minerals are notorious for forming filamentous and tubular structures through entirely non-biological processes. When iron-rich fluids precipitate minerals, the crystals can grow in elongated forms. When fluids flow through porous rock, they can leave behind tube-like mineral deposits in the voids. When rocks are deformed, iron minerals can be stretched into filaments. Without carbon, without a precise age, without additional context, the Nuvuagituk structures are ambiguous at best. They might be fossils. They're consistent with fossils, but they could just as easily be mineral artifacts. And in science, when you're making an extraordinary claim, the oldest fossils on Earth might be isn't good enough. So the consensus remains. The Apex Chert microfossils at 3.465 billion years old are the oldest widely accepted fossils on Earth. Everything older is controversial, suggestive, debated. We're at the edge of the knowable. But let's step back and think about what we've learned. What this entire quest has revealed about life, Earth, and the nature of scientific discovery itself. We started with Mary Anning, standing on a storm-lashed beach in 1833, staring at the eye socket of an ichthyosaur, a creature 200 million years old. And we thought that was ancient, unimaginably ancient. And it is, by human standards, by historical standards. But now we've traveled back in time 17 times further, to 3.5 billion years ago, to a world so alien, so different from anything we know, that it might as well be another planet. No oxygen in the air, no ozone layer protecting the surface from radiation, no soil, no plants, no animals, no fungi, just bacteria and archaea living in shallow seas and around volcanic vents, slowly, patiently transforming the planet's chemistry. And we've pushed even further, to 3.7 billion years in Greenland, 3.8, 4.1 billion years in a single zircon crystal. We're reaching back to the very origin of life itself, to the moment when chemistry became biology, when the first molecule figured out how to make copies of itself, when the first cell membrane enclosed the first self-replicating system, when life, in the most basic sense, began. And we've found evidence, not conclusive, not always clear, but evidence, fossils, chemical signatures, stromatolites, microbial mats, traces of organisms that lived and died billions of years ago, leaving faint echoes in the rocks. Against all odds, against the relentless destruction of geological time, these echoes have survived, waiting for us to find them. But we've also learned something else, something humbling. We've learned that there's a limit to what we can know, a boundary beyond which the evidence has been destroyed, the rocks melted, the fossils erased. No matter how hard we look, no matter what technology we develop, we can't see past a certain point, can't prove what happened in the first few 100 million years of Earth's history, because the record simply doesn't exist anymore. The oldest rocks on Earth are around 4.03 billion years old, tiny fragments from the Acasta gneiss in northern Canada. But they're so metamorphosed, so altered, that they contain no fossils, no organic matter, nothing biological, just isotopic hints, ambiguous and controversial. Beyond that, nothing. The first 500 million years of Earth's history are lost. The Hadean Eon. We know it happened. We can model it. We can infer it from other evidence, but we can't observe it directly. The rocks are gone. So when we ask, what is the oldest fossil on Earth? We're really asking, how far back can we see before the record goes dark? And the answer, for now, is 3.465 billion years. The Apex Chert, Bill Schopf's discovery, vindicated after decades of controversy. But there's a twist to this story, one final revelation that changes everything. Because the oldest fossil on Earth might not be on Earth at all. It might be on Mars. Think about it. Mars today is cold, dry, dead. But 4 billion years ago, Mars was warm. It had oceans, lakes, rivers, a thick atmosphere, volcanic activity, all the conditions that gave rise to life on Earth. And because Mars is smaller than Earth, it cooled faster, which means it became habitable earlier. By the time Earth's surface solidified, Mars might have already had liquid water for hundreds of millions of years. If life arose fast on Earth, it almost certainly arose on Mars too. And if it did, there should be fossils, microbial mats, stromatolites, maybe more, preserved in ancient Martian rocks from 4 billion years ago. And here's the kicker. Those fossils would be better preserved than anything on Earth. Because Mars doesn't have plate tectonics. The crust is geologically dead. Rocks that formed 4 billion years ago are still sitting on the surface, largely unaltered, not subducted, not metamorphosed, not melted and recycled, just sitting there, waiting. NASA's Perseverance rover is currently exploring Jezero crater on Mars, a crater that was once a lake, 3.5 billion years ago, exactly the same age as the Apex Chert. The rover is collecting samples of ancient lake sediments. And in the early 2030s, if all goes according to plan, those samples will be returned to Earth, and scientists will examine them for fossils. If we find microbial fossils in those Martian rocks, it will be the greatest scientific discovery in human history. Proof that life arose twice, independently, in the same solar system. Proof that life is common in the universe. And quite possibly, proof that life on Mars is older and better preserved than life on Earth. The oldest fossil might not be in the Australian outback. It might be in a sample tube on a rover waiting to come home. And beyond Mars, the icy moons of Jupiter and Saturn, Europa, and Enceladus both have subsurface oceans, liquid water in contact with rocky cores, hydrothermal vents, chemical energy, everything life needs. If life exists there, and we have no reason to think it doesn't, there will be fossils, microbes living around vents, dying, sinking, being preserved in seafloor sediments. Except those oceans have no plate tectonics either, no recycling. Sediments just accumulate, layer upon layer, for billions of years. The deepest layers could contain fossils from the very beginning of the solar system, 4.5 billion years old, older than anything on Earth or Mars. A complete fossil record from the origin of life to the present, preserved in eternal darkness beneath kilometers of ice. We won't reach those fossils in our lifetimes, maybe not in our grandchildren's lifetimes, but they're there, waiting, and someday someone will find them. So when we ask, what is the oldest fossil, we're asking the wrong question. The right question is, where should we look? And the answer is, everywhere. Earth, Mars, Europa, Enceladus, Titan, exoplanets. Everywhere there's water and chemistry and time. Because life, we've learned, arises fast, spreads quickly, persists tenaciously, and leaves traces, faint, fragile, easily destroyed traces, but traces nonetheless. Written in carbon isotopes and microscopic filaments and layered domes of stone, echoes of organisms that lived and died billions of years ago, in places we can barely imagine, under conditions we can barely understand. Mary Anning found an eye socket. We found carbon filaments in desert rocks. The next generation might find microbial mats on Mars. The generation after that, stromatolites on Europa. Each discovery pushing the boundary of the knowable. Each fossil a message from deep time. But let's return to Earth, to the present, to the question we started with. What is the oldest fossil on Earth? The answer is the Apex Chert microfossils, 3,465,000,000 years old. Filamentous structures of organic carbon, isotopically light, morphologically diverse, consistent with biological origin. Found in Western Australia by Bill Schopf in the early 1990s. Challenged by Martin Brasier for over a decade. Finally vindicated by isotopic analysis in 2018. They're not the oldest hints of life. The Jack Hills Zircon suggests life at 4.1 billion years. The isotopes stromatolites hint at 3.7. The Nouveau-Argituc tubes claim even older. But none of those are accepted as definitive fossils. None have the combination of morphology, chemistry, and context that the Apex Chert provides. So for now, 3.465 billion years is the answer. That's as far back as we can see with confidence. That's where the fossil record begins. Everything before that is inference, speculation, educated guessing based on incomplete evidence. And honestly, that's okay. Because 3.5 billion years is extraordinary. It tells us that life arose within the first billion years of Earth's existence. That by the time the oldest surviving rocks formed, life had already diversified into multiple forms. That the origin of life wasn't a slow, gradual process stretching over billions of years. It was fast, explosive, almost immediate in geological terms. And that changes how we think about our place in the cosmos. We're not a miracle, not a 1 in a trillion fluke. We're what happens when chemistry runs its course on a wet, rocky planet. We're inevitable, or at least highly probable, which means we're probably not alone. But here's the sobering part. We've reached the limit, the hard edge of what fossilization can preserve, what rocks can survive, what time hasn't destroyed. We can speculate about life 4 billion years ago, but we can't prove it, can't see it directly. The evidence is gone, melted back into the mantle, eroded into dust, lost forever. So when we look at those Apex Chert filaments under a microscope, we're not just looking at fossils. We're looking at the edge of history, the boundary between the knowable and the unknowable. Beyond this point, the past becomes mythology. We can tell stories about what might have happened, but we can't know. Not really, unless we look elsewhere. Mars, Europa, places where the geological record is more complete, where 4 billion-year-old rocks still exist, unaltered, waiting to tell their stories, where the fossil record might extend all the way back to the origin of life itself. That's the future. That's where the next great discoveries will be made. Not in the deserts of Australia or the mountains of Greenland, but on other worlds, in other oceans, under other skies. Mary Anning braved storms to find ichthyosaurs. Bill Schopf endured the Australian outback to find bacteria. The next generation will brave the vacuum of space to find life on Mars, on Europa, on worlds we haven't even discovered yet. And when they do, when they bring back samples from ancient Martian lakes or drill through the ice of Europa, when they find fossils 4 billion years old, perfectly preserved, with cellular structures and DNA and proteins intact, the oldest fossil on Earth will become a footnote, a local record, impressive for its time. But superseded, because Earth, it turns out, isn't the best place to look for the oldest fossils. It's just the easiest, the most accessible, the only place we can reach right now. But it's not the archive we need, not the complete record, just a fragment, a tantalizing glimpse, enough to tell us that life is ancient and resilient and probably common, but not enough to show us how it began. For that, we need to go elsewhere. And we will, because that's what humans do. We search, we explore, we push boundaries. We refuse to accept that the past is unknowable just because it's difficult to know. Mary Anning understood this. She risked her life to recover fossils because she knew they mattered, because she understood, even if the scientific establishment of her time didn't, that the past holds lessons for the present. That understanding where we came from helps us understand where we're going. Bill Schopf understood this too. He spent decades studying rocks that most scientists considered too old, too altered, too hopeless to contain the fossils. He endured criticism, skepticism, outright hostility. But he kept working, kept searching, kept refining his methods, because he knew the fossils were there. And he was right. And somewhere right now, a graduate student is preparing thin sections of rocks from Greenland, or analyzing isotopes from Mars, or designing a mission to drill through Europa's ice. And they don't know it yet. But they're about to make the next great discovery, the next breakthrough, the next revelation that pushes the boundary of the knowable just a little bit further. That's how science works. Not through sudden epiphanies or lone geniuses, but through patient accumulation of evidence, through incremental improvements in technology, through careful, rigorous, skeptical examination of every claim, through debate and controversy and eventual consensus, through generations of scientists building on each other's work. Mary Anning found fossils in limestone. Bill Schopf found fossils in chert. The next generation will find fossils in basalt, on Mars, and the generation after that will find them in ice, on Europa. Each discovery made possible by the ones that came before, each answer raising new questions, each boundary pushed revealing new horizons. So what is the oldest fossil on Earth? 3.465 billion years. The Apex Chert. Filaments of ancient bacteria preserved against impossible odds, waiting 3.5 billion years to be discovered by a species that wouldn't exist for 3.5 billion years. But what is the oldest fossil in the universe? We don't know yet, but we're looking, and we will find it, because that's what we do. We look back, we dig deep, we refuse to accept that the past is lost just because it's distant. And every time we find a fossil, every time we push the record back another million years, another 100 million years, we're doing something profound. We're recovering our history, our origins, our place in the cosmos. We're reading the book of life, page by ancient page. And the oldest pages, the ones we're reading now, tell a story of a planet barely cooled from its birth, of oceans heavy with iron, of skies thick with methane, of tiny cells clinging to volcanic vents and shallow seafloors, building, growing, evolving, transforming a hostile world into a habitable one. Those cells are our ancestors, Every living thing on Earth today, from bacteria to whales to humans, descends from those first organisms. We carry their genes, their metabolic pathways, their evolutionary legacy. We are made of star stuff, yes. But more than that, we are made of them. Those first cells. Those 3.5 billion-year-old bacteria, invisible to the naked eye, fragile beyond imagining, but tough enough to survive the worst the universe could throw at them. They survived asteroid impacts, volcanic eruptions, climate shifts that froze entire oceans, oxygenation that poisoned the atmosphere, mass extinctions that killed 99% of species. They survived, adapted, thrived, and passed their genes down through billions of generations until, eventually, impossibly, some of their descendants developed brains, consciousness, the ability to look back across deep time and ask, where did we come from? And now we know. We came from them, from those filaments in the Apex Chert, from those mats in the Dresser Formation, from those cells around hydrothermal vents 4 billion years ago. We are the universe waking up, becoming aware of itself, looking back at its own history. And the oldest fossils, those faint traces in ancient stone, are mirrors, showing us not just what life was, but what life is. Resilient, adaptable, tenacious, capable of surviving anything. Given time, given opportunity, given a planet with water and chemistry and energy, life finds a way. It always has. For 3.5 billion years at least. Probably longer. Maybe much longer. And it always will. Long after we're gone. Long after our fossils are the ancient ones. Long after some future species stands where we stand now, looking back, asking the same questions. What is the oldest fossil? How far back can we see? What came before? The answers will be different. The boundaries will have moved, the records more complete, but the questions will remain. Because they're not really questions about the past. They're questions about ourselves, about what it means to be alive, to be aware, to be part of something vast and ancient and ongoing. Mary Anning stood on that beach in 1833, holding an ichthyosaur skull, and she asked, "What is this? Where did it come from? What happened to it?" We're still asking those questions, still standing on beaches, metaphorically, holding fossils, wondering, still pushing back into deep time, still searching for origins. Still refusing to accept ignorance when knowledge, however difficult to obtain, is possible. And the oldest fossils, 3.465 billion years old, are answers. Not complete answers, not final answers, but answers nonetheless. They tell us life is old, life is tough, life is persistent, and life, given the right conditions, arises fast and spreads far. That's the message from deep time, written in carbon filaments, preserved in Australian stone, waiting 3.5 billion years to be read. And now we've read it, understood it, and we're ready for the next message. From Mars, from Europa, from worlds we haven't imagined yet. The Book of Life has many pages. We've read the oldest ones Earth has to offer. Now we're reaching for the rest of the book, the chapters written on other worlds, in other oceans, under other stars. And when we find them, when we read them, we'll finally understand not just where we came from, but why we're here and where we're going. Because the past, it turns out, is prologue. The story of life is still being written, and we're part of it. Characters and authors both. The oldest fossil on Earth is 3,465 million years old. But the story it tells is timeless, infinite, ongoing, and it's our story, all of ours, from those first cells to us and beyond. That's what Mary Anning discovered, standing on that storm-battered beach, that the past isn't dead, it's alive in us, in our DNA, in our bones, in our curiosity, in our refusal to stop searching, stop questioning, stop wondering. And that's what the oldest fossils teach us. Not just scientific facts about ancient bacteria, but something deeper, more profound, that we're connected to the deep past, to the distant future, to life everywhere it exists in the cosmos. We're not alone in space. We're not alone in time. We're part of a continuity stretching back billions of years and forward billions more. And the fossils, those fragile traces in ancient stone, are the evidence, the proof, the gift from deep time, telling us, you came from this, you are this, you will become this. Life, enduring, transforming, eternal.